What exactly occurs in the body when you get cancer? What is cancer? So I would say cancer is essentially a story of DNA damage and incorrect correction. Now, as you know, DNA is the genetic information in our cell. There are about 2 meters long strands of DNA in our cells, in every single cell, 2 meters, which is highly wrapped around and packaged through this amazing system. So there's multiple layers of, that thread packaged around proteins, those proteins then wrapped around themselves, then wrapped and wrapped. There's about 6 levels. I've got these boxes of further reading in green on most slides, so if you're interested, you can go and read more on that. And so this genetic information, is encoded in the sequence of the DNA. Here you have, we've got 4 main bases, and I don't know if you've done this yet in GCSE, but you'll definitely learn about it in more detail at A-level biology, where you've got 4 bases which encode the sequence of the DNA, and based on the sequence, there are different amino acids that are put and tied together to make different kinds of proteins. And so the sequence is very precise and it's very, very important. Now, if you get a damage to this original sequence, which can happen through a lot of different causes, sunlight, for example, they say UV light, that causes actual damage. Other environmental factors can cause that. What you eat can cause that. General wear and tear in the cells can cause that. So when you actually get a damage to one of these bases, usually what happens is that our DNA repair system within the cell is really, really good at actually putting back the right base, but sometimes it can actually put in incorrect bases, and that's what's called a mutation. Now, mutations may lead to then abnormal proteins being produced because the code has now changed. What can also happen is sometimes you still get the normal protein being produced, but in abnormal quantities. Now, these proteins can be important for the function of the cell, for example, helping the cell to divide. But what happens is if a mutation isn't corrected and you're getting abnormal levels of that protein, you can actually start getting cells that behave very abnormally. So going back to our example of the protein that helps cells divide, if you get too much of that protein, the cells are going to end up dividing more. And there's this thing called the Knudson's two-hit hypothesis, which essentially says that only 2 mutations are enough to make a cell cancerous. And so here I show you a number of normal cells, and just imagine if one of them gets this mutation, 2 mutations, and starts behaving abnormally, and it starts dividing uncontrollably. What will happen is it ends up taking over and starts dividing way more than it should, and you end up getting a lump where you have excessive division of cells, and you have too many cells, and that lump is essentially the lump of cancer, and a tumor has formed. So, one bad cell can lead to an entire tumor, so it is very scary, actually. So is there a cure for cancer? There are myths out there that say, oh, there's a cure for cancer already found and scientists and pharma companies are hiding it. Or people say, oh, there's going to be one miracle cure for cancer. Both of these are incorrect, because cancer, in fact, is not just one disease. You think about all the tissues and the different types of tissues that are in your body, lung tissue, kidney tissue, etc. Each of these can develop cancer, and each of these cancers is going to be different. Well, on top of that, as you saw before, 2 mutations are enough to lead to cancer, which means that there are so many genes in your body that could potentially get mutated, and each of those different types of mutations would lead to a different type of cancer. And so there isn't ever going to be one cure for cancer. There are cures for cancers of certain types. For example, childhood lymphomas are cured very well. Certain types of breast cancer can be treated really well, and there are other cancers that have no treatment or are very poorly treated. So there's no single answer to a cure for cancer. So why is cancer so hard to treat? I did go a bit into how every single cancer type could be very different and it would require different treatments. But the more we study cancer in detail in the laboratories, we realize that actually it's even more complicated than that, and it's just really, really tricky and challenging because there is something called tumor heterogeneity and cancer evolution. So I want you to remember this cell, and you saw that one cell could end up leading to a tumor, but not only that, I will show you that branch tree thing again. Today seems to be a talk of these branch trees, and you might study this in your A-level biology, looking at the evolution of species and natural selection. And very interestingly, a very similar process happens within cancer cells. So for example, this one cell that I showed you in the previous slide, that will go on over time and it will acquire more and more mutations, and those, as it divides and becomes daughter cells, those daughter cells then divide, becoming more daughter cells. They themselves will acquire more and more mutations because a lot is already going wrong in that cell. They are already very prone to more and more DNA damage and incorrect repair. That, for example, as it gets here, this cell, when it divides into these two daughter cells, this one might still be the same, but this one will acquire more mutations. Then as it divides, it might acquire more mutations. Then you have this daughter cell. At this point, it might acquire even more mutations. And so what ends up happening is you get a very heterogeneous tumor at the end, which looks like that, and a bunch of so many different cells. And then it comes to treating it, you think, oh my gosh, what do you target? Do you target the green cells? Because that's going to require different medication. The blue cells might require something totally different. The pink cells will require something totally different. And so that is why cancer is becoming so hard to treat, the more we find out about it. And so it might all sound very dismal, but is there a way ahead, and what can we do now that we know this? One of the main things that we can do, and something that my research focuses on and I'm really passionate about, is actually early diagnosis of cancer. So it's, instead of giving the cancer so much time to become so heterogeneous and get to that stage where it's so hard to treat, if we can find it early enough, then we can actually treat it much better. So early diagnosis is something that a lot of research is currently focused on because a lot of studies have shown that 1 in 3 cancer deaths could be prevented if you diagnose cancers early. If you look at this picture over here from Cancer Research UK, it shows you that 81% of cancer of people could survive up to 10 years or longer if their cancer was diagnosed early, whereas if the cancer is diagnosed at stage 3 or 4, then there's only a 26% chance of surviving 10 years or longer. So this is obviously very important, and currently why we struggle to find cancer early is that to diagnose the cancer early, it needs to be big enough for us to be able to see it using imaging machines like radio or MRI and CT scans. But usually when the cancer is big enough to be seen, it's big enough, it's already too big and very hard to treat. So how can we actually diagnose cancers earlier then? Before I talk to you about that, and that is something my research focuses on, I want to tell you a little background story of how I got into what I got into. And this is a story of an accidental discovery in yeast. You heard that, yeast, to find a cure for cancer. So before I joined the Humphrey Lab in Oxford, they had been working on yeast cells, and they did a little drug screen where they actually tried different types of yeast with different deletions in certain genes, and they would look at which drugs actually kill the yeast. And so they serendipitously found that there was this yeast which had a mutation in a gene called SED2, And these yeast were dying when given a certain drug called WEE1 inhibitors. Turns out that this gene, the SET2 gene, is actually mutated in a lot of human cancers. So we have got the same gene as yeast because of evolution of species. We share a lot of genes with other organisms. And so a PhD student in the lab tried out this drug in human cancer cells and showed that actually we could kill cancer cells that had this mutation and this change inside them. And then they tried it out in mouse tumors and also showed that giving this drug actually shrank the tumors much more. So this was then taken forward into clinical trials, and that's currently being tested. Now, where I come in now with my research is I'm trying to find a way to diagnose these types of cancers earlier using blood tests, and a very cool thing called DNA methylation, which I'm going to talk to you about. So what is DNA methylation? Probably the coolest thing in all of biology, but I am a bit biased because I do work on it. So you can think of it as a little flag or a mark or a tag on top of certain sites on your DNA. Essentially, it's a methyl group, a CH3 group. For you, those of you that like chemistry, you can go check out what DNA methylation, the mark, looks like. But essentially, it can, so many things that are around you in the environment, or what you eat, or your lifestyle can actually end up tagging your DNA very differently. And this can have an impact of what happens inside your cell, because DNA methylation can actually regulate the production of proteins. And so it's a very interesting case of a link between your environment and what you eat and what's happening inside your cell at a very molecular level. Now, a very cool thing about DNA methylation, and the reason it can be used and has such big potential for the early diagnosis of cancer, is that cancer cells and your body's own cells, in fact, release a lot of DNA into the blood. So you've got this circulating DNA, it's called cell-free DNA or circulating tumor DNA, and you can read more about that if you like. And you can actually look at the different patterns on the circulating tumor DNA, and based on a blood test, it could potentially tell you what is happening inside your cells, inside your body. So this is great for early diagnosis, and because a very small tumor will still be exuding this DNA, so it doesn't need to be big enough to be seen. You also don't need to do a surgical process to remove that tissue and look at what it looks like and what the cancer is like. So it's really exciting, and there's a lot of work if you want to go research on it, just DNA methylation biomarkers for cancer, etc. There's a whole field. And another cool thing for the people who are interested in mathematics, DNA methylation, the way it's read, because a single DNA site can either be methylated or unmethylated, so you get a 1 or a 0, and then you can actually take an average of lots of different DNA molecules at the same site. And so you can take an average and you can actually get an actual number. And what can we do with numbers is that we can actually put them into equations. And there's this thing called a Horvath age clock, or a DNA methylation age clock, where these people looked at sort of like 353 CpG sites, which are these DNA methylation sites in the DNA, and they calculated people's age based on DNA methylation. And I've actually tried it myself. Not on my own DNA, but using data online, I actually looked at DNA methylation levels, and then I ended up calculating people's age to between 3 years, and it was very accurate. So it's really cool to be able to use DNA methylation to predict a person's age and things like that. So go check that out. It's a very interesting thing. So what do I do with DNA methylation? I'm going to explain that using juggling. This was a presentation I did for the DPhil day here, and I ended up winning first prize, and the BBC actually ended up interviewing me on this. So crazy things can happen when you work in science. So I'll play that for you, and I hope you enjoy it.
Hi everyone, I'm Hira and I'm doing a DPhil in oncology, and I'm going to try and explain my research using juggling. So I want to introduce DNA methylation to you. So it's an epigenetic mark, and it comprises a methyl group, CH3 group, which is put on top of different bases in your DNA, and usually these are cytosines. And it's a very important mark because it can actually regulate gene expression. And if you take this as an example of DNA methylation, and blue being the gene expression, sometimes there's a positive correlation. So the more methylation you get, the more the gene is switched on and expressed, and vice versa. And sometimes there's a positive correlation, but it's just an association, and it's not methylation actually regulating gene expression. And other times, it's actually negatively correlated. So the more methylation you have, the less gene expression you have. A really cool application of DNA methylation has been to make age clocks, because methylation varies with age, and so a 20-year-old might have this pattern, whereas a 40-year-old might have this pattern, and an 80-year-old might have this pattern. And so they've actually made age clocks which can calculate your age looking at your DNA methylation patterns to within ±3 years, which is actually quite cool. Also, DNA methylation changes can alter risk for cancer, and these changes start occurring very early on in cancer. And so if we could make a blood test using DNA methylation changes, we can potentially diagnose cancers much earlier and also non-invasively. So this is what I'm trying to do. I'm trying to develop a blood test to diagnose specific types of cancers that have lost a histone mark, and these cancers can be more aggressive, patients have worse prognosis, and also we've shown in our lab that we can very selectively target these types of cancers with certain inhibitors that are being tested in clinical trials. So if we could have a blood test to diagnose who has these cancers, then we'd be able to better select patients that will actually respond to the therapy and benefit from it. And so in my first year, I've been looking at DNA methylation patterns across the genome to try and figure out which sites are different enough to allow us to classify the cancers into the ones that have lost the histone mark and the ones that haven't lost the histone mark. So I've looked at methylation array data at 450,000 sites across the genome, and what I found is that some sites have exactly the same pattern when it comes to the cancer that has lost the histone mark and that hasn't lost the histone mark. And so these are the sites that won't help us classify anything. So there are some sites which show a different methylation pattern when it comes to cancers that have lost the histone mark versus the ones that haven't lost a histone mark. But then sometimes these sites actually have the same exact pattern in normal tissue, and you don't want that. You don't want to diagnose normal tissue. You want to diagnose a very specific type of cancer. So again, these sites excluded. Then there are some sites which show a very kind of chaotic pattern that changes, and you don't know if it's changing with the loss of the histone mark or if it's some other environmental factor or age, and these can be quite confounding factors and hard to tease out. And again, these are the sites that are not trustworthy, and you don't want to use these. Then there are other DNA methylation sites where the pattern is different, but the change is just so subtle that you're better off not using these sites. So this is what I've been trying to do in my first year, trying to pick out sites that are significantly different in methylation in the cancers that have lost the histone mark, that have big enough changes in methylation, that are not impacted by confounding factors, that are not varying with age or other things that might affect methylation, and trying out different cancers and trying to figure out which sites are common, because we want to develop a pan-cancer biomarker that would work in any cancer type. And I found about 1,000 sites. The next step would be to try and narrow this down to as few sites as possible, but a good enough test with a high specificity and sensitivity, because the fewer the sites, the cheaper the blood test will be. So that is what I'm planning to do, and thank you for listening and watching, and I hope you enjoyed the juggling. I welcome any questions.
So I hope you found that interesting, and that tells you a bit about my research and what I do here. at Oxford. If you have any questions, or if you would like to learn more, or if you just want me to give you some resources, then please feel free to email me, and I'll be happy to get back to you. So yeah, I look forward to hearing from you, and enjoy biology, enjoy maths. I hope this sort of like inspired you a bit to learn more. Just enjoy learning, and it will get you places. So take care and have a good rest of your day. Bye.